Hello friends, this is the second video of enzymology. So let's begin and let's begin from enzyme specificity. What do you mean by enzyme specificity? Enzymes are highly specific in their action when compared with the chemical catalyst. The occurrence of thousands of enzymes in the biological systems might be due to the specific nature of the enzymes. There are three types of enzyme specificity are well organized, well recognized. First is stereospecificity, second is reaction specificity, third is substrate specificity. Specificity is a characteristic property of the active site. Now let's know about stereospecificity or optical specificity. Stereoisomers are the compounds which have the same molecular formula but differ in their structural configuration. The enzymes act only on one isomer and therefore exhibit stereoisomerism. Example: L-amino acid oxidase and D-amino acid oxidase act on L and D amino acids respectively. Hexokinase act on D-hexoses, glucokinase on D-glucose, and amylase acts on alpha-glucosidic linkase. Cellulose cleavage cleaves beta-glycosidic bonds. Stereospecificity is explained by considering three distinct regions of substrate molecule, specifically binding with three complementary regions on the surface of the enzyme. This class of enzymes belonging to isomerase do not exhibit stereospecificity since they are specialized in the interconversion of isomers. Now let's know about reaction specificity. The same substrate can undergo different types of reactions, each catalyzed by a separate enzyme, and this is referred to as reaction specificity. An amino acid can undergo transamination, oxidative deamination, decarboxylation, racemization, etc. The enzymes, however, are different for each of these reactions. Next, let's know about third point, that is substrate specificity. The substrate specificity varies from the enzyme to enzymes. It may be either absolute, relative or broad. Now let's know about absolute substrate specificity. Certain enzymes act only on one substrate. Example: glucokinase acts on glucose to give glucose 6-phosphate. Urease cleaves urea to ammonia and carbon dioxide. Now let's know about relative substrate specificity. Some enzymes act on structurally related substances. This in turn may be dependent on the specific group or a bond present. The action of trypsin and chymotrypsin is a good example for group specificity. Trypsin hydrolyzes peptide linkage involving arginine or lysine. Chymotrypsin cleaves peptide bonds attached to aromatic amino acids. Example of bond specificity: glucosidases acting on glucosidic bonds of carbohydrates, lipases cleaving ester bonds of lipids. Now let's know about broad specificity. Some enzymes act on closely related substrate, which is commonly known as broad substrate specificity. Example: hexokinase acts on glucose, fructose, mannose and glucosamine and not on galactose. Now let's know about coenzymes. Many enzymes require certain non-protein small additional factors, collectively referred to as cofactors, for catalysis. The cofactors may be organic or inorganic in nature. The non-protein organic, low molecular weight and dialysable substance associated with enzyme function is known as coenzyme. The functional enzyme is referred to as holoenzyme, which is made up of protein part and non-protein part. The term activator represents the inorganic cofactor like calcium, iron, Mg2+, Ca2+, Mg2+, Mn2+. etc. necessary to enhance enzyme activity. Now let's know about coenzymes are second substrates. Coenzymes are often regarded as the second substrates or co-substrates since they have affinity with the enzyme comparable with that of the substrates. Coenzymes undergo alterations during the enzymatic reactions, which are later regenerated. This is in contrast to the substrate which is converted to the product. Coenzymes participate in various reactions involving transfer of atoms or groups like hydrogen, aldehyde, keto, amino, acyl, methyl, carbon dioxide, etc. Coenzymes play a decisive role in enzyme function. Now let's know about coenzyme from beta B complex vitamins. Most of the coenzymes are the derivatives of the water soluble B complex vitamins. In fact, the biochemical functions of B complex vitamins are exerted through their respective coenzymes. The chapter on vitamins gives the details in structural and summary function of the coenzymes. We can study there. Okay, so let's know about non-vitamin coenzymes. Not all coenzymes are vitamin derivatives. There are some other organic substances which have no relation with vitamins but function as coenzymes. They may be considered as non-vitamin coenzymes, example ATP, CDP, UDP, etc. Nucleotide and coenzymes. Some of the coenzymes possess nitrogenous base, sugar and phosphate. Such coenzymes are therefore regarded as nucleotides NAD+, NADP+, FMN, FAD, coenzyme A, UDPH, UDPG, etc. Now let's know about coenzyme do not decide enzyme specificity. A particular coenzyme may participate in catalytic reactions along with different enzymes. For instance, NAD+ acts as a coenzyme for lactate dehydrogenase and alcohol dehydrogenase. In both the enzymatic reactions, NAD+ is involved in hydrogen transfer. The specificity of the enzyme is mostly dependent on the apoenzyme and not on the coenzyme. Now let's know about mechanism of enzyme action. Catalysis is the prime function of enzymes. The nature of catalysis taking place in the biological system is similar to that of non-biological catalysis. For any chemical reaction to occur, the reactants have to be an active state or a transition state. Now let's know about enzymes lower activation energy. The energy required by the reactants to undergo the reaction is known as activation energy. The reactants when heated attain the activation energy. The enzyme the catalyst reduces the activation energy and this causes the reaction to proceed at a lower temperature. Enzymes do not alter the equilibrium constants, they only enhance the velocity of the reaction. The role of a catalyst or an enzyme is comparable with a tunnel made in a mountain to reduce the barrier. The enzyme lowers energy barrier of reactants, thereby making the reaction go faster. The enzymes reduce the activation energy of the reactants in such a way that all the biological systems occur at body temperature below 40 degrees centigrade. Now let's know about enzyme substrate complex formation. The prime requisite for enzyme catalyst is that the substrate must combine with the enzyme at the active site to form enzyme substrate complex, which ultimately results in the product formation. Now let's know about some theories on this enzyme substrate complex. First is lock and key model or lock and key theory, or we can say Fischer's template theory. It is otherwise known as lock and key theory is otherwise known as Fischer's template theory. According to this model, the structure or conformation of the enzyme is rigid. The substrate fits to the binding site just as a key fits into the proper lock or a hand into the proper glove. Thus, the active site of an enzyme is a rigid and pre-shaped template where only a specific substrate can bind. This model does not give any scope for the flexible nature of enzymes. Hence, the model totally fails to explain many facts of enzymatic reactions, the most important being the effect of allosteric modulators. Now, let's know about induced fit theory or Kosland's model. Kosland in 1958 proposed a more acceptable and realistic model for enzyme substrate complex formation. As per this model, the active site is not rigid and pre-set. The essential feature of the substrate binding site are present at the nascent active site. The interaction of the substrate with the enzyme induces a fit or a conformation change in the enzyme, resulting in the formation of a strong substrate binding site. Further, due to the induced fit, the appropriate amino acids of the enzyme are repositioned to form the active site and bring about the catalysis. Induced fit model has sufficient experimental evidence from the X-ray diffraction studies. Kosland's model also explains the action of allosteric modulators and competitive inhibition on enzymes. Now let's know about substrate strain theory. In this model, the substrate is strained due to the reduced conformation change in the enzyme. It is also possible that when a substrate binds to the performed active site, the enzyme induces a stain to the substrate. The stain substrate leads to the formation of product. The concept of substrate strain explains the role of enzyme in increasing the rate of reaction. In fact, a combination of the reduced fit model with substrate strain is considered to be operative in enzymatic reaction. Now let's know about mechanism of enzyme catalyst. The formation of an enzyme substrate complex is very crucial for the catalysis or to occur. It is estimated that an enzyme catalyzed reaction proceeds 10 to the power 6 to 10 to the power 12 times faster than a non-catalyzed reaction. It is worthwhile to briefly understand the ways and means through which the catalytic process takes place, leading to the product formation. The enhancement is in the rate of the reaction is mainly due to the four processes. First is acid-base catalysis, second is substrate strain, third is covalent catalysis, fourth is entropy effects. Now let's know about diagnostic importance of enzymes. Estimation of enzyme activities in biological fluids is of great clinical importance. Enzymes in the circulation are divided into two groups. First is plasma specific or plasma functional enzymes. Certain enzymes are normally present in the plasma and they have specific functions to perform. Generally, these enzyme activities are higher in plasma than in the tissues. They are mostly synthesized in the liver and enter the circulation. Example, lipoprotein, lipase, plasmin, thrombin, cholinesterase, ceruloplasmin, etc. Impairment in liver function or genetic disorders often leads to a fall in the activities of plasma functional enzymes. Example, deficiencies in ceruloplasmin in Wilson's disease. Now let's know about non-plasma specific or plasma non-functional enzymes. These enzymes are either totally absent or present at a low concentration in plasma compared to their levels found in the tissues. The digestive enzymes of the gastrointestinal tract, example amylase, pepsin, trypsin, lipase, etc., present in the plasma are known as secretory enzymes. All the other plasma enzymes associated with metabolism of the cells are collectively referred to as constitutive enzymes. Example, lactate dehydrogenase, transaminases, acids, and alkaline phosphatases, creatine phosphokinase. Estimation. Now let's know about isoenzymes. The multiple forms of an enzyme catalyzing the same reaction are isoenzymes or isozymes. They, however, differ in their physical and chemical properties, which include the structure, electrophoretic and immunological properties, Km and Vmax values, pH optimum, relative susceptibility to inhibitors and degree of denaturation. Example, lactate dehydrogenase, 5 isozymes, creatine kinase, 3 isozyme. Finally finished. Any questions regarding this, you can ask me in the comment box, and subscribe my channels to know more updates. Thank you.